Good afternoon and welcome to the e-seminar series 2021 of the European Society of Gene and Cell Therapy. My name is Hildegard Büning. I'm Professor of Infection Biology at the Gene Transfer at Hannover Medical School and the current president of the ESGCT. The ESGCT, or European Society of Gene and Cell Therapy, is a non-profit organization promoting fundamental and clinical research in gene therapy, cell therapy, and in vaccine development. Creating a platform for scientific networking and exchange, as well as education, are parts of our mission. We therefore launched the ESGCT e-seminar series. The first 3 seminars are dedicated to genome editing and were kindly sponsored by Intellia Therapeutics. So far, we have covered the topic how it all started with a seminar of Professor Rege of the Vilnius University, who introduced us into the topic and shared his latest results. Last week, this was followed by a seminar of Professor Toni Katomen of the University Hospital Freiburg. In his seminar, we discussed the topic safety. Now, it's my pleasure to introduce you to Dr. Laura Sepp-Lorenzino, the Executive Vice President and CSO of Intellia Therapeutics. Dr. Sepp-Lorenzino studied biochemistry at the University of Buenos Aires, Argentina, and received both her master's and Ph.D. in biochemistry from the New York University. She is an expert in nucleic acid therapies, as well as in oncology drug discovery and development. Both expertise that she acquired when leading the cancer research department at the Merck West Point and working as an assistant attending molecular biologist at the Memorial Sloan Kettering Cancer Center. As I said, she is now Chief Scientific Officer at Intellia Therapeutics, and thus oversees all platform and pipeline research activities. Today, she will speak about what is next in CRISPR, and we are very much looking forward to hear her vision.
Thank you very much, and hello, everybody. Special thank you to ESGCT and Hildegard for this great introduction. I really appreciate this opportunity to talk to you about Intellia and how we're using CRISPR-Cas9 as a transformational tool to precisely manipulate the genome to develop potentially curative therapies. First, please make note of our forward-looking statement as a public company. So here is an outline of my talk. First is going to be an introduction to our modular genome editing platform. Second, I'll give a short description of what's CRISPR-Cas9 and how we're using it. And then in the last part of the talk, it will be the application of the technology for in vivo knockout and insertion, as well as for ex vivo cell engineering. So Intellia Therapeutics, which where I work as the CSO, is a leading gene editing company. We're based in Cambridge, in Massachusetts, in the US. And it was co-founded by leaders in the field, including Jennifer Doudna, that, as you know, won the Nobel Prize last year for her discovery of CRISPR, along with Emmanuel Charpentier. So the mission of the company is to transform the lives of people living with severe diseases by developing curative genome editing treatments. And this is really what keeps us focused on advancing safe and effective therapeutic solutions for patients. So central to this mission is CRISPR-Cas9 as the tool that we're using to specifically, precisely introduce therapeutic changes in the genome. These changes can lead to inactivating or knocking out disease-causing genes, repairing genetic errors, or restoring the expression of proteins that were either missing or proteins that have a new desired functionality. And we do that by insertion. Importantly, these genetic changes are permanent in the cell that was edited, and this offers the potential for curative, long-lasting treatments. And I'll hope to show you evidence towards reinforcing that point. So CRISPR-Cas9 is part of this modular platform. Here we have gene editing tools, not just one, but the tools that are required to introduce different types of edits, the best tool for the job, as well as the delivery solutions that we can apply in vivo and ex vivo to make CRISPR into therapies. For genetic diseases shown on the left of the slide, CRISPR-Cas9 is the therapy. And here we deliver it systemically via non-viral lipid nanoparticle delivery system as our first delivery solution. For cancers and for autoimmune diseases, here we utilize CRISPR as a tool to engineer cells outside the body, either from the patient or from a donor. We rewire those cells with new instructions, and then upon infusion into the patients, those cells become a living drug. So here is a snapshot of our current development pipeline that is the result of deploying this modular platform with editing and delivery solution. Shown on the top, our in vivo pipeline with CRISPR being the therapy. We're in the clinic with Intella 2001 for transthyretin amyloidosis. Here we're knocking out a gene in the liver, a disease-causing gene that's made by the liver cells. This is a program in phase 1 that we are partnered with Regeneron, with Intellia being the leading party. And that's going to be followed this year with Intella 2002, which is also a knockout program where we're knocking out a protein that's called prekallikrein that is key in mediating hereditary angioedema. And this is a wholly owned program. And that's followed by a number of knockout and insertion programs. I'll tell you a bit more about those programs in the next few slides. On the bottom, we're showing our ex vivo pipeline where CRISPR creates the therapy. We're in the clinic with OTQ 923 for sickle cell disease with Novartis, with them being the lead party. And that is taking hematopoietic stem cells and editing ex vivo and infusing them in the patient for sickle cell disease. Wholly owned is the next program, that's Intella 51. Here we're developing an autologous TCR or T cell receptor T cell therapy for acute myelogenous leukemia. And this is followed by a number of programs in research and preclinical state. So moving on to the second part, what's CRISPR-Cas9, what's the mechanism, and how do we apply it? So CRISPR-Cas9 is a programmable endonuclease system. It's originally from bacteria, where it plays an adaptive immune system mechanism. And it was quickly repurposed, for the use of precise editing in eukaryotes. The components are a CRISPR-associated protein, or Cas, and that is the programmable endonuclease that binds to a guide RNA, which is a short single-strand RNA molecule. And that RNA determines the genomic location for Cas to bind to, for Cas to become activated and introduce a gene edit. The excitement around this technology is that it's simple to deploy the Cas enzyme to different genomic locations just by changing the guide RNA. And as I will show you, the only thing you change is the address part of the guide RNA. And that really made it, a very efficient, simple, scalable system. And there is an explosion of utility, right, of this, or demonstration of utility of this system, just, look at PubMed and, thousands and thousands of applications. So the breakthrough work, right, as I was telling you, was recognized by the 2020 Nobel Prize in Chemistry for Jennifer Doudna and sharper TA, but also there is, the other founders of the company have contributed significantly to advancing this field. So now let's go a bit deeper on the mechanism. And there are a number of factors that contribute to the specificity of the edits introduced by Cas, and I think it's important for us to review. First, the Cas enzyme is inactive when not bound to the guide RNA. So it's not going to be introducing any edits unless it's bound to the guide RNA. The guide RNA shown here in teal, it binds to the Cas protein by a region called the scaffold that's constant between all the different guides. And once that it binds to Cas, it forms a complex called a ribonucleoprotein complex, or RNP. And once that it binds, the complex undergoes a number of conformational changes that makes it competent to scan the DNA, identify the target sequence, open up the DNA, and then become catalytically active to introduce the break in the DNA. There are a number of Cas systems from different bacteria, so they change slightly on, where they cut, how they cut, and the structure of the guide RNA, but these are general rules that apply across all of the different cases. The specific target recognition depends on two factors. First is on the presence of a short sequence called the protospacer adjacent motif, or PAN, that is specific to each Cas and is adjacent to the target site. And then there is the actual target site that is recognized by a variable sequence in the guide RNA that recognizes the target site by Watson-Crick complementarity. So once that all of these happen, as I was saying before, in the case of SP Cas9 or Streptococcus pyogenes Cas9, you introduce a double strand break because there are two catalytic sites on opposite side of the strands. So once that there is a double strand break, that's all that the Cas system do. So what happens next is that the host cell, the eukaryotic cells, has very active DNA repair mechanism that will immediately recognize that double strand break or that nick and get into action. There are two main repair mechanisms. The prevalent one is called non-homologous end-joining, or NHJ. That's active on all cells, dividing and non-dividing cells. And this immediately causes that break to rejoin. Now, it's not a very precise mechanism, so you end up generating some small insertions and deletions called indels that, depending on where you select the edit to occur, can result on, for example, loss of expression of that gene. On the right side, there's the other mechanism that's called homology-directed repair, or HDR, that relies on the presence of a donor DNA or a source of template for either correction or for insertion of a particular sequence. This HDR mechanism is only active on dividing cells. So, if you're going in vivo, there are, few cell types in which there is active division and active ability to use the HDR system. So keeping this in mind, how now do we develop CRISPR-Cas9 drug candidates for therapies, right? So because so much depends on the guide RNA being able to determine the precise location of the edit, is very important that there is a workflow to carefully and thoroughly identify and deeply characterize those guide RNAs so they can only introduce the edit in the on target with minimal to no off-target activity as undesired feature. For this purpose, we developed a comprehensive workflow that applies a combination of both in silico and empirical approaches, and we use genome-wide assays and next-generation sequencing, or NGS, to identify and verify the most active, precise, specific, clean guide RNAs for a driven gene target. And over the next 4 slides, I'll show you how we do that. So the starting point is to select the therapeutic gene, obviously, what's your target. And then for that gene, we identify all of the potential target sites that are going to be determined by the PAM sequence for the Cas enzyme that you're using. We then rank order all of those guys based on several criteria, including avoiding repetitive or variable regions or SNPs across the population. We like, not always possible, but we like to have cross-species homology, so we can use safety species, full homology guides between the safety species and the human. That really facilitates preclinical work. And then, of course, that there are no off-target sites. And because the target site is determined by sequence complementarity, we can use bioinformatic tools and in silico prediction algorithms as the first step to identify these potential guide RNAs. Then we make them. We synthesize a guide RNA synthetically. We use solid phase synthesis in a 96 well plate format. And we have the ability to introduce chemical modifications, the same ones we're going to be using in the actual drug. Then we do primary and secondary screens of the therapeutically relevant, with the therapeutically relevant cell types. For example, for our liver program, we use primary human hepatocytes from multiple donors. So these assays here allows us to rank order the guides first in a primary in vitro screen, single dose point, and we just rank order the guide RNAs, from best to worst. Then we take the best guides, and then we do an in vitro dose response, and that gives us, better clarity on the IC50 for introducing the edit on target, and we use NGS methods to do this. And then we further select those guides, and we do an in vitro functional readout where we correlate the percentage of gene editing with the pharmacodynamic consequence. And that, for example, for a knockout approach will be decrease in the expression of the mRNA and of the protein. Okay, so once that, we have a number of guides that have really good, potent, on target, we need to move on to do a comprehensive analysis of potential off targets. So for that, we also rely on in silico prediction to look at all the potential off targets. We put those on the list. Then we use empirical approaches such as a biochemical off-target discovery method called site-seq, where we expose chromatin to an RNP with the different guides, and then we look at all the places where it cuts. We take those places, we add it to the list. And then we generate panels of NGS reagents, so we can go through the list one by one and do those response curves and understanding whether we see off targets at those potential sites. And if we do, we make those response curves to understand the difference between the on target and the off target. Of course, the goal is to select guides that do not have any detectable off-target cutting at a supra pharmacological dose of what you intend to use in the clinic. So with that, then you end up with, very few guides that you want to, that are your candidates. And we use a set of complementary now technologies to characterize any potential DNA structural variants. For that, we use short and long read sequencing, as well as we use direct visualization of the genome to look for any chromosomal rearrangement or any, any potential issue that we want to avoid. Okay, so that's how we do it, right? So now how do we apply it? And first, let's talk about, in vivo, right? How do we develop CRISPR in vivo drugs? So again, CRISPR is the drug. We're going to be giving the gene editing machinery systemically by IV administration to a patient. And the goal here is that those cells are going to be edited permanently, and that we're going to do that in such a way that for the patient, we could have a potential curative therapy after a single dose. So central to how we deliver the CRISPR-Cas9 in vivo is the use of a non-viral delivery system. And for this, we use lipid nanoparticles. So these are self-assembled particles. Basically, they resemble dietary fat. And this is how the body looks at them and distributes them and process them. So the LNPs are synthetic, they are self-assembled. They can, so the way we use them is that we co-formulate a messenger RNA that codes for the Cas9 protein. In the same particle, we put the target guide RNA. So what happens inside the cell? The mRNA will make protein. That protein will have the guide RNA present at the same time. These guys will bind, will go to the nucleus, will find the particular specific target location and make the edits. What we like about LNP delivery, these are clinically proven for hepatocytes in the liver. They have a large cargo capacity, unlike AAV, for example, where you're restricted on what you can package. Another difference from viral delivery system is that, this is transient expression. All of the components have been designed to be biodegradable, and actually it's only expressed for a few hours, and within a day or two, all of the components are eliminated. There is no, limiting immunogenicity. There is no pre-existing or de novo immunogenicity that will limit dosing or potentially redosing. They are well tolerated. We understand what are the class toxicities, and these are monitorable, reversible, dose dependent. So there is, safety window that we can apply. And, importantly, these are synthetic, chemical in nature. So it's easy to scale up and manufacture in a reproducible manner. So how are we using gene editing in vivo? So we can use it to knock out genes, to insert genes, and to do both. And we call that consecutive editing. And I'll tell you more of each as we cover the next slides. So starting with our knockout approach. So here our lead candidate is called Intella, and it's in phase 1 clinical studies in the UK and New Zealand for the treatment of all clinical manifestations of a disease called Transthyretin amyloidosis, or ATTR. So this is a progressive disease that is caused by the accumulation of a liver hepatocyte-made protein called transthyretin that is secreted into serum. And in the disease, it accumulates in multiple organs, it deposits, particularly in nerves, in hearts, in kidneys and eye. And the manifestation of that is polyneuropathy and cardiomyopathy. And some patients, or most of the patients, have both. It's a mixed phenotype. The disease could be hereditary, where there are a number of mutations, but can also be wild type. And the numbers range from 50,000 from hereditary to up to 500,000 for people with a wild type form of the disease. So our goal is to use a lipid nanoparticle delivery system to take CRISPR to the hepatocytes in the liver and make a knockout edit on the TTR gene. So now that TTR gene is no longer producing TTR, no more. So you're shutting down the faucet of TTR. And the goal is that, we can do this one and have profound degree of TTR knockdown, so you can stop the progression of the disease, but perhaps, and we're looking to reverse the disease. And because the expectation is that this is going to be durable, that these patients will have only one course of treatment and be potentially cured. So what's the data in support of this? So this slide shows the validation of the approach in non-human primates. And I want to highlight that for most, oligo and nucleic acid therapies, non-human primates have proven to be a predictive species for human. So here we give a single intravenous intravenous administration of our lead LNP with the CRISPR machinery that will target the TTR gene in the liver. Here, because these are cynomolgus monkeys, the TTR guide RNA recognizes the cyno gene. So as you could see here, if we follow TTR protein in serum as a percentage of the baseline, and this is the control levels, once that you give a single dose of the gene editing machinery and you shut down the expression of that gene, you see a very rapid decline in the production and in the, the presence of TTR in the serum. And because that gene is shut off, once that you reach a nadir, that nadir is stable. And here I want to highlight the x-axis. We're looking at 1 year after a single dose. And as you could see, the levels are flat. What's highlighted here in light blue is 60% knockdown has been shown by other mechanisms of reducing TTR protein levels by knocking down the expression of the mRNA with antisense and RNAi. They show that 60% is the threshold where you begin to see clinical benefit. Anything greater than that is expected to have greater clinical benefit. And as you could see here with 2 dose levels, we're achieving what we believe are going to be very therapeutically relevant to patients at doses that we believe are going to be safe and tolerated in humans. So NTLA-21 is being evaluated in a global phase 1 study. The study is a 2-part open label multi-center study with 2 sites currently in the UK and in New Zealand. There are 2 parts of the study. It's a single ascending dose with 4 cohorts, followed by a single dose expansion cohort at the optimal biological dose. So, we're very excited about this because it's the first, systemic administration of CRISPR machinery. And, we'll keep everybody informed when we're ready to release data on the study. So, to wrap up, on NTLA-21, very exciting. And the goals for this year, of course, is to continue to enroll patients, to demonstrate that CRISPR works in humans, that is safe, and that particular for TTR, that we can achieve what we believe are going to be very therapeutically relevant doses in a safe manner. So moving on to the next program, and this builds on the modularity of the liver editing system. Once that you figure out how you deliver something to hepatocytes to knock out the gene, then what's the next indication that will follow the same approach? And for this is we're going for a disease called hereditary angioedema. These patients have recurring severe swelling in different parts of the body. Is caused by a loss of expression of a gene called C1 inhibitor. And here we're aiming to knock out the prekallikrein gene to regain control of the cascade of events that leads to these unexpected edema events. So again, the potential is one and done. Minimize the production of kallikrein, and by producing a stable reduction in kallikrein, we're aiming to have continuous prophylaxis of these patients so they will not have these attacks. So here we're building on the modularity, as I was saying. So same LNP as we use for 2001, same mRNA, same structure of the guide. The only thing that changes is the target site in the guide RNA, which is 20 nucleotides on the 5' end of the guide RNA. As I just showed you in non-human primates for TTR, if we do the same experiment now targeting kallikrein in the hepatocytes in the liver, once that you shut down expression of that gene by a single dose of our CRISPR machinery, those levels remain flat. And here is 1 year after a single dose, 3 dose levels, all of them as shown for TTR, are way within the therapeutically relevant level of kallikrein activity reduction. And here we're using data from an antibody that we know that 60% reduction in kallikrein, leads to good prophylaxis. What we're offering is that, here is one and done. And because there is no variability on PK, once that you reach that nadir, the patients are going to be stably protected. So the goal for this program is to submit an IND in the second half, IND or IND equivalent in the second half of the year. So I've been making the point that, here we're looking at potentially one and done, long lasting. We waited a year in monkey for 2 different studies, but a year is not a lifetime. So what we're trying to do is to find orthogonal approaches that can add additional information on this durability question. So what we did here is we use a partial hepatectomy study where you introduce an edit, you remove 2/3 of the liver, and then you wait for liver regeneration and ask the question, did the edits remain or were they lost with the rapid proliferation? And as shown on this slide, we have 2 groups, control animals in teal, TTR knockout animals in blue. As shown by the middle panels, the LNP administration showed profound reduction of the TTR by knockout. So after we confirmed that we had edited the liver, we removed 2/3 of the liver, waited until the liver came back to normal size, and then we evaluated whether the edits were maintained. And as shown by the third set of bars here, post regeneration, all of those edits were maintained, right? So this, supports that we are editing cells that will contribute to the homeostasis of the organ, and that over time they're supposed to, to maintain the edit for a long time. So layering in more complexity now that we can do knockouts in the liver, what else can we do? And And the next level of complexity is insertion. So what do we do here? We use the LNP platform again to deliver the CRISPR-Cas9 components to make a cut on the DNA in a transient manner. We don't want Cas lingering around. And then what we use is a promoterless AAV as the means for delivering of the gene of interest for insertion into that cut. The first application of this is for hemophilia B, and people at ESGCT are very well familiar with what the disease is and the need for constant and good level of expression of factor 9 for hemophilia B. So how do we do the insertion? So here we're targeting the site of insertion as intron 1 of the albumin locus in the hepatocytes in the liver. We choose albumin to take advantage of the strength of the promoter. It's a protein that's self-regulated, so there is very good expression and control. And we also take advantage of the secretion signal of albumin for secreted genes such as factor 9. We're using AAV9 to deliver a bidirectional promoterless construct. So the AAV by itself will not express anything because it's no promoter. We are relying on a mechanism of insertion that is a blunt insertion, and that's why we're using a bidirectional template. So it could be inserted and expressed on either orientation. And once that expression happens, we're going to have, there is going to be from exon 1 splicing into a splice acceptor next to the gene of interest, and there is going to be production of that mRNA leading to production of factor 9. So shown here is data from mice where we looked at how many of the cells in the hepatocytes were positive for insertion, and we could see about 50% of them expressing the inserted gene. That results in stable expression of factor 9 detectable in blood. This is a study done in mice a year after a single dose, very stable expression. We have different levers that we use to modulate how much insertion we want to get. That is the exact sequence in the intron 1, and we've seen that depending on the location, we could have more or less expression. The other levers are the dose of the AAV for the template for insertion and the dose of the LNP for the CRISPR machinery. This is data we've shared last year on the levels of factor 9 achieved in non-human primates using two different guide RNAs with fixed AAV and LNP doses. And depending on where we choose to insert, we could go from normal levels to much greater levels. Not that we intend to use these greater levels, but gives you an idea of the capacity of the system. As I showed you before, long-lasting, we use the same experiment of partial hepatectomy to interrogate duration. And here we compare side-by-side a CRISPR-mediated gene editing approach to a traditional gene therapy AAV with promoter for expression of factor 9. So we took two cohorts of animals. Both of them, so in teal, are animals that had traditional AAV gene therapy. In blue, CRISPR insertion for factor 9. Here is 100% of circulating factor 9 levels in a mouse. We did partial hepatectomy, removed 2/3 of the liver, waited for the liver to regrow, sacrificed the animals at 2 time points. And what we've seen is that whereas the AAV expression was lost after the rapid proliferation of the liver, which makes sense due to the episomal nature of the gene of interest. By contrast, the DNA that was stably inserted by CRISPR was retained, and there was no loss at 54 or 85 days after the insertion. So, we're quite excited about, the durability and the opportunity. Quickly covering how we're taking these from hemophilia. And by the way, hemophilia A and B, factor 8 and factor 9, are being pursued in collaboration with Regeneron being the lead party. The next program for us is alpha-1 antitrypsin deficiency. That's a wholly owned Intellia program. So here is it's a different type of disease, because here you have a toxic gene in the liver that should have been secreted. So the disease manifestations are, in minority of the patients, you see liver disease for accumulation of this toxic protein. But the more prevalent manifestation of the disease is because the protein is not secreted, is not active systemically, and these patients present with emphysema because there is a lack of alpha-1 antitrypsin deficiency to counteract the action of neutrophil elastase, particularly in lung. So here, what we want to do is, one of the options that, we're pursuing is consecutive editing. So what this means is, go into the liver, knock out the toxic gene, but now you need to reinsert the wild type gene. So you will follow with an insertion approach. And this is the proof of concept in rodents. So here are rodents that express the mutant form of alpha-1 antitrypsin called the Z allele. So first we came in with an LNP that has a guide RNA that recognizes the Z allele, and we can, as shown here, knock out the expression of that mutant protein. And once that's knocked out, it's knocked out. Here we're looking at 15 weeks after the intervention. And what we did is then we followed up 3 weeks after the initial treatment with a second treatment, where now we use an LNP with the same Cas, but now the guide determines a cut for insertion in the albumin intron 1. And we use a promoterless AAV to take the alpha-1 antitrypsin gene. And as you could see, within 2 weeks, you begin to clearly see production of the wild type protein. And once that's produced, that is stable. At the alpha-1 meeting in December, we show data now in non-human primates with what we believe are unprecedented levels of expression of alpha-1 in non-human primates. So here we have animals, and we use the insertion approach for the wild type AAT. And again, within 3-4 weeks of after the insertion, you begin to achieve normal, or you achieve normal levels of alpha-1 antitrypsin. And just for reference, here I'm showing what the therapeutically relevant levels of alpha-1 achieved with enzyme replacement are, but it's widely agreed that those are likely insufficient as a curative approach. So we're very excited about this program, and aiming to continue the development, and we'll tell you more as we progress the program. So a couple of things on insertion. High levels of expression, right? So I'm being able to, depending on the protein we want to express, to using different doses of AAV and LNP and the specific site of insertion to modulate expression. And then on the right side is the durable protein expression, right? So here, the goal is that these changes are going to offer a benefit over traditional gene therapy, including the ability to intervene really early in disease, including pediatrics. So switching now gears to the other side of the company, and this is how we're using CRISPR-Cas9 as a tool to now take cells, rewire them with all the necessary instructions for them to become living drugs in a patient. There we go. So our initial focus is for immuno-oncology and autoimmune disease. Like we did for in vivo, here we built a modular platform where we have a cell engineering platform that allows us to do highly efficient editing. And importantly, we do the editing in a sequential manner. So we make one edit, let it fix, wait, and do the next edit. And this, we believe, is a significant advantage and advance on the use of this technology. And I'll show you one slide on why I believe that. And then we can deploy this technology in different cell types with different immuno-oncology or autoimmunity targeting modality. Those could be T-cell receptors or CARs or universal CARs. And because we can introduce multiple edits, we not only do insert the targeting modality, but we can make all the necessary changes to, again, rewire that cell. So why I believe our proprietary method is superior? Well, so it allows us to have cells that will not, what most people use in the field is, follow manufacturer's instructions for, different electroporetic technologies. One of the issues with that is a significant loss in viability, and those cells, they don't expand really well. And this is shown here, untreated T cells versus cells that, followed standard manufacturing practices. What we've done is try to understand every single step so our cells have high viability and expansion potential. But because we need to introduce multiple edits, we don't want to cut the genome all at once because you could have multiple rearrangements between all of those free ends. So by doing it in a sequential manner, we can completely eliminate the risk of translocations and any chromosomal rearrangements that not only reduces the yield of your cell product, but could have potential safety consequences. Our first application is Intella 51 for acute myelogenous leukemia. So here we're using a TCR or T-cell receptor mediated approach for immuno-oncology. The T-cell receptor recognizes a peptide derived from a tumor antigen called Wilms tumor 1 or WT1. WT1 is a cancer antigen. For AML, the relevance is that this is expressed in all the different types of AML, regardless of the driving mutation. And even when there is minimal residual disease, those AML blasts retain expression of WT1. So it's a really good target for us to develop an immuno-oncology T-cell approach for. So what we do here is we take T cells from the patient. So Intella 51 is an autologous system. We remove the endogenous TCR on those T cells, and in its place, we introduce the WT1 specific TCR. And why this is important? Because if you don't remove the endogenous TCR, you end up, as shown here, with a mixed cell product. The cells are going to have the endogenous TCR, the transgenic TCR, as well as a mixture of both. And that reduces yield and also produces uncertainty with respect to safety, because you are going to have heterodimers with unknown recognition properties, and that's a safety risk. The way we do this, we are aiming to have an estimated vein-to-vein time in these patients of 3 weeks. And we have shared this data at ASH in December, showing that in a model, in vivo model of acute myelogenous leukemia, where we're using patient-derived AML cells, not cell lines, we could see a very nice control of the disease. Shown here in red, there is complete tumor stasis as compared to a control untreated group, or a group in which we made T cells with an irrelevant T cell receptor. So we're very excited about this program. We moving this to submit an IND or IND equivalent in the middle of this year. And to wrap up, I hope I provided evidence of how we look to use CRISPR as the foundation for generating a pipeline of differentiated products. Today, we're focused on genetic diseases and immuno-oncology and autoimmunity. We, in the research team, we continue to work towards building that modular platform to unlock the full potential of the technology. We're looking at different LNPs that could make it to different organs, particularly bone marrow, to do, for example, sickle cell disease and beta thalassemia editing in vivo without the need of a transplant. And for ex vivo, we're pushing towards autoimmunity, regenerative medicines, and different mechanisms for ex vivo approach. And with that, I just want to leave this slide with our upcoming milestones, and really appreciate everybody listening to me. We're hiring, so please visit Intellia, tx.com careers, and reach out to me or any of my colleagues. I'm happy to answer any questions.
Thank you very much, Laura, for this fantastic talk and for sharing all these data. We have received already a lot of questions, and I just would like also again to let the audience know that they can type in their questions, which I will then tell you. Before I use the first question from the chat, let me ask one by myself. So you were showing nicely that you had this stable modification in the liver. And the question is, what kind of cells do you target? Of course, hepatocytes, but I mean, they showed stable modification. So what kind of cells are you targeting there? Any clue?
Yeah, so we're looking at hepatocytes. That's the main target cell. The reason for this is you need, for the insertion to work, you need to have two things, transfecting and transducing at the same time, right? So for LNP, we have is an ApoE-mediated mechanism. LDLR is a mechanism of uptake. And most of at the doses used, most of the hepatocytes in the liver take on the CRISPR machinery. AAV8, right? So some of those hepatocytes are going to be co-transduced now with AAV8, and you need to have those two things to have effective insertion. As part of the program, of course, we're doing wide biodistribution studies and looking for insertion. But the expectation, and the data we have so far supports, the hepatocyte as the main source.
So that means the stability is based on the amount of hepatocytes you are targeting?
Yeah, so the level of expression is based on the number of hepatocytes, again, in which we successfully deliver the LNP and the AAV donor.
Yeah, it was just regarding your Hepatectomy experiment, yeah, where you really saw that despite doing this, you had a stable expression. So right. If I got you right, it's due to the amount of modification you can achieve in your liver.
So that, and the ability to get to cells that will contribute to that regeneration. As you know, there are a number of theories on whether, the prevailing theory said there is hepatocytes being re-engaged as part of keeping the homeostasis of the liver rather than stem cell niche. But whether it's one or the other, with our methods, we're getting to both.
Okay, thanks. So the first question is concerning the pump. So if you don't have a pump sequence nearby, so what are you doing?
So if you don't have a pump sequence, you are, that Cas9 will not work for that gene. Luckily, Cas9, up to now, we have not found a case where we have not been able to identify the guide. We do have other enzymes with different pumps, and that allows us to, cast a wider net to get the appropriate guides.
Okay, Wound Boring from UMC Utrecht in the Netherlands is asking, what are your thoughts on integrase deficient lentivirus or AAV particles to deliver the CRISPR-Cas in vivo?
So, for me, what's really important with CRISPR-Cas, because Cas is bacterial in origin, right? I want to make sure that the Cas expression is transient. So, and LNPs as a delivery mechanism gives you transient expression. The limitation is that not all organs will be amenable to an LNP delivery system. So viral delivery systems do come into play, and I think they're important. But what's important is to have self-regulated or self-inactivating systems. So you only have transient expression of Cas. And why do I say that? Because there is pre-existing immunity, against Cas. There are T cells. So if you have constant expression of Cas, for example, by a constant expression from an AAV system, you're going to be presenting that Cas protein, and you run the risk that you will eliminate by immunity the cells that you edited, because they're going to be presenting Cas. So I think that it's important that we have really good transient self-inactivating models or systems for viral delivery.
Thanks. Louis Kono from AstraZeneca is asking, do you detect any LNP-related toxicity in your studies?
So for LNPs as a class, they do have a number of class toxicities. These are infusion reactions. And then you could see transient transaminases. So, and this has been seen by all LNPs. The goal, right, is to make sure that you have a therapeutic window where you don't see any of those at the intended therapeutic dose, right? Or so if you push the dose with LNPs, of course, you are going to see ALTs and, other consequences. When I was showing the data, we do have a therapeutic window that, we intend to demonstrate in humans.
The next question are concerning the gRNA. So at the gRNA selection stage, if your best gRNA have imperfect homology between human and your model system, how does that complicate the further development?
So yeah, and we've encountered that as well. So ideally you want to have perfect homology, right? So you could do all non-human primate safety and exacerbated pharmacology, and that simplifies things. If you don't, so if your guide RNA has some activity in the monkey, you can still do it, right? Because you could get exaggerated pharmacology with a monkey-specific guide, and then use your clinical candidate as part of your safety activity, safety studies. If they're completely different, then you and this is a dialogue you need to have with the agencies, right, on what they're going to be looking. But you may need to have different tools to demonstrate pharmacology, and and then your clinical candidate for the IND enabling studies. But for everybody, these are conversations you're going to have with your regulatory agencies.
Leah is asking, how do you purify the LNPs and what kind of quality assessments do you do? So purity, correct mRNA packaging, etc. And maybe I add, can you also comment on the stability of your LNPs?
Yeah. So, and I'm not sure how much we have shared as to the release criteria, but yes, I mean, you're looking, for size, polydispersity, concentration, charge, zeta potential, all of that is part of a package, including the potency, sterility, all of that. So, and these are really widely accepted release criteria. I want to highlight, Onpattro, for example, is an approved drug based on LNP. So, these criteria is clearly described and characterized. With regards to with stability. So we have developed frozen formulations that, they have really long stability, as frozen stocks.
Thanks. Kuna Das is asking about the efficacy of your hybrid therapy of LNP and AAV.
The efficacy.
Right. Yeah.
So we've shown it now for, factor 9 and for alpha-1 antitrypsin. Similar to what we've shown before for TTR and HAE on the knockout, the platform really, determines the performance. So we're really happy that the levels of expression we achieve for factor 9, are similar to the levels of expression we can achieve with alpha-1 antitrypsin, meaning that once that you set the dose, of the LNP and the AAV, basically the body and the same site for insertion, right? It's being driven by the albumin promoter. You minimize the variation from program to program, right, for expression. So I think it's the gene of interest becomes the gene of interest, right? And you don't see much variability between one and the other.
Thanks. Carsten Lederer says, great talk and data.
Thank you.
Elsewhere, there have been serious adverse events, possibly related to myoablation in both the CTX001 and Therapeutics trials. Regarding therapy in HSPCs and for your own portfolio, do you see a way to reduce or avoid adverse events caused by myoablation for HSPCs-based gene editing therapies?
So we have, and I cannot comment on our HSC because this is led by Novartis. They're the primary lead here, but in our own research, we have not seen any effects that will be, deleterious. So I think that some of that is, cell culture and different cell type conditions on how the cell editing is done. So again, it's the editing, whether you're using a plasmid or using a transient system, all of these things come into, bearing for the outcome of the on-target and potential deleterious effects. So keep that in mind.
And the next question, AGTC Ventures. Do you think there is a good approach to ex vivo AAT cell engineering therapy versus gene therapy with AAV and LNPs given hepato uptake of cell therapy?
So I'm not sure I'm getting the question. So you use cell therapy for liver?
This is what I understood, yeah.
So there is a lot of potential on using, gene editing for regenerative medicines. I'm not sure I would use it for AAT when we have other solutions, but certainly, there are cell therapies that you could use not just for immuno-oncology or immunity, but, regenerative medicine, whether it's, diabetes or some of the CNS diseases. There, what's important is having a true allogeneic approach. And actually, I didn't have time to cover this, but we'll be presenting on what we believe will be a universal, robust allo approach in the coming weeks.
Thanks. I don't see any further questions. Let me check. No. So again, thank you very much for this wonderful talk. It was really, really inspiring. And I also would like again to thank Intellia for sponsoring this mini-series. And I would like also to thank all the audience for this nice discussion. And would like to announce that our next series will then start in April. So have a nice day and thanks again. Bye-bye.
Thank you.